Our next speaker is Zach Bateson. He's a research scientist at the National Agricultural Genotyping Center. He's really a perfect complement to the talk that you just heard from Doreen Ware, because he essentially takes the information from plant genomes and transforms that information into molecular diagnostics for pathogens and pests. This also takes advantage of his formal training, which is in population genetics. Now, I have to tell you that when he's not busy developing diagnostics, you can find Zach out on the North Dakota prairie trying to identify all the plants and animals he finds there with his family. He's also an amateur zookeeper whose house is home to 17 snakes, 2 lizards, 1 frog, 3 salamanders, 1 tarantula, and a cat. So with that interesting tidbit, I'd like to welcome Zach to our talk for today.
Thanks for that great introduction. I don't know if it's good that everyone knows I have a mini zoo at home or not, but that's okay. So yeah, thanks for coming to my talk. It's great to be here. I'm tasked with telling the group about some of the diagnostic technology that we're using at the National Ag Genotyping Center. I know I have an audience of students as well as teachers. And so for the first part of my outline, I really want to talk about kind of my research background, my career pathway that I took, because it has nothing really to do with agriculture. I shifted into agriculture later on in my career. After I kind of convince you of that and how I can transfer the genetic tools that I learned in my graduate program to agriculture, I'm going to talk a little bit about the diagnostic biotech that we use, real shallow dive into quantitative polymerase chain reaction, or qPCR. There might be a lot of you that actively use it or have heard of it, especially with the coronavirus. It's the same technology that they use to amplify that virus. So I'll kind of cover that a little bit. And then I'll jump into some of the current research projects we have going on at NAGC. So I grew up in northern Minnesota. My backyard was essentially a big forest. So I got lost all the time as a kid out in the woods. And that's really where I got my interest in the living world. Of course, my mother would get upset. I'd come home late after dark. So I always was tending to get in trouble getting lost in the woods. But that's really what inspired me to pursue a degree in biology. And in one particular memory that I have, this is my childhood driveway. Just beyond the woods right here was this small pool. And this pool had tons of creatures in it, hundreds, maybe thousands. And when I was about 7 or 8, I'd go to that pool and just stare at it for a couple hours every day. And I was just fascinated by these creatures in there. Over the summer, though, the water would go down. And I'd race off to the spigot to grab more water to keep my little creatures alive and increasing in numbers. Well, these are the creatures that I was taking care of. I'll give you a second to take a look at that. Maybe some of you know what they are. They're mosquito larvae. So I used to be a mosquito breeder when I was a child. Probably something you don't want to admit, but that's really where I got my passion for this type of work. I'm happy to say I'm a retired breeder now, though. So along with having that interest as a child and having great junior high and high school biology teachers, I got really interested and continued into a biology career. So I got my undergraduate at North Dakota State University in Fargo, where I'm actually located right now. And my interest kind of evolved into reptiles. And so I took care of the reptile room as an undergrad. I'm happy to say, even though these pictures are of me holding alligators, I still can count to 10 with both hands. But that's really where I got my interest in those organisms. And then wanted to kind of explore a career in research dealing with reptiles. And so for my master's degree, I looked at a particular research question: do females have genetically diverse clutches in a state endangered species called the five-line skink? And so you might be wondering, well, why is that important? Well, a lot of these populations that I studied were on the periphery of their range. Because they were on the periphery of their range, there's harsh environments going on, and generally you want to have a diverse, genetically diverse population. For females, it makes sense that they want a genetically diverse offspring for this purpose. And so the interesting thing about this lizard is that it has maternal care, so the female protects the eggs. And that allowed us to capture both the females and the potential offspring to get at this question. And so you might be wondering, well, how do you figure out the genetics of the offspring? And you do it pretty much the same way you do it in people. And so you do paternity analysis using PCR and looking at microsatellite or simple sequence repeat markers. So these neutral markers across the genome to kind of get at this question. And so here's just kind of a basic output from this analysis. So here's the mother's genotype at a single locus. I highlighted both of her alleles with this green. And then each one of these lines is the genotype at this marker for each of her babies or each of her offspring. And so if you just run your eyes down, you'll notice that every baby has the green allele, but then there's these other alleles. Those are going to be the ones from the father, the paternal allele. And so what you'll notice by the color, I've highlighted the different ones. There's 3 altogether. That's telling us that at least 2 males sired this clutch, because a male can only provide 2 alleles to its offspring. And so this is kind of the way we got to this question: do females have genetically diverse clutches? And the answer was yes. In fact, 60% of the clutches we looked at had multiple paternity. So using kind of this groundwork genetic tool, I moved into my PhD research. And I looked at a different organism. And I actually kind of went into, instead of reproductive biology, I went into conservation genetics, using the exact same tools that I've been using. And so this question was, can translocated birds boost genetic diversity in an endangered population? And so for this, what I did is I looked at this research project that was going on where there's an endangered population of this bird called the greater prairie chicken in the state of Wisconsin. It has been in decline for decades, and they wanted to boost the population genetically. And so what they did is they translocated birds from Minnesota, which has a much larger population, over to the Wisconsin population to try to boost that genetic diversity. And so in 2006 through 2009, they brought birds over, females, from Minnesota over to Wisconsin in a fashion that kind of looked something like this. That animation I'm most proud about out of all of my slides of this talk. So the question then, did these translocated birds boost genetic diversity in that Wisconsin population? And so how we did that is we tracked the genes from Minnesota to see if they could be found in Wisconsin, just like tracking those birds. And so what I'm showing you here is just a simple output of one of the markers we looked at. We looked at a mitochondrial marker called D-loop. And so what essentially you do is you count up the number of haplotypes, so the unique, you can think of them as alleles in the population. And so on the y-axis here, I have the number of haplotypes, and then the x-axis is the groups that we looked at. And so these Minnesota females that were brought over, they had 20 haplotypes altogether. So a very robust haplotype diversity in those that were brought over. Before this translocation happened in Wisconsin, pre-Wisconsin, there was only 6 haplotypes. So definitely a reduced amount of genetic diversity. However, after the translocation, this is in 2011, when I sampled the Wisconsin or post-Wisconsin population, there was a climb up to 10 haplotypes. And within those 10 haplotypes, we know that 4 of them came from Minnesota. Now, there's a little bit more to the story, but what's interesting is we have historic data from back in 1952 from mummified wings that were found up in a house. It's a whole story in itself. But with that box of wings, we could look at the genetic diversity historically in the Wisconsin population and see that we boosted genetic diversity, at least at this marker, to near historic levels. So from these two stories I just talked about, the graduate work in skinks and birds, these genetic tools that I learned throughout this graduate program, I was able to shift into my career, because my career now is in livestock, honeybees, and plant genetics, mostly in pathogens within this group of organisms. So a little bit of background about the company I work for, the National Ag Genotyping Center. So we started in 2016. There's 3 of us, soon to be 4 on staff. We have over 40 years of experience with this type of DNA analysis and diagnostics. We're an ISO accredited lab, meaning that we have protocols and safety measures and quantity assurance measures for all of our tests. One thing that's important that Dr. Ware mentioned too is that this is a highly collaborative project. So all these projects I'm going to be talking about, we have collaborators that are both public and private sector that have been important throughout kind of the last 4 or 5 years that we've been around. So this is just kind of a summary of the group that we have collaborated with honeybees for the most part. So what really motivates us as a lab is bridging this gap between research and practical applications in biotech. There's a lot of awesome research that happens at the academic level, at universities, at the USDA ARS, and we want to try to find ways to bring that to the producers themselves. There's a lot of diagnostic tests, for example, that are used across agriculture that can be used to help growers, but generally once a project's done in academia, sometimes they get pushed off to the side. And so we kind of help resurrect those to find applications for them. And our kind of area or discipline is diagnostics, because compared to clinical sciences and humans, even in livestock, if you're talking cattle and horses, diagnostic is lagging, especially in plant pathology. And so we want to try to help that industry out, because we know pests and pathogens are some of the top threats to crops and food supplies. So I wanted to kind of go through our diagnostic, our PCR diagnostic work that we do. And so kind of stay kind of, sorry, my computer just shut down. Some of the processes that we use. So we use, like I said, polymerase chain reaction, or PCR, for all of our diagnostic tests. Essentially, if you're not familiar with it, it's just a molecular copier for copying small segments of DNA or RNA, depending on what you're looking at. And so we use it as a diagnostic tool. So you can go from small quantities of DNA or RNA to large enough copies so we can look at it using a gel or using light sensitive equipment. That all kind of depends on the assay. So just kind of a broad base overview of what we do and kind of the behind the scenes. Essentially, the first step is to identify the genetic target. This target could be a pathogen. It could be a single trait that you're interested in a crop or in honeybees or something like that. That can be a hard step to come across. And then once you discover it or identify it, then you find unique genomic regions. So we rely a lot on bioinformatics to help us find these unique regions. And just to kind of give you a brief overview of that, this bottom panel here is showing you DNA sequence alignment. And so I have DNA sequences on top of each other from non-targets, which are these top 2 sequences. The target sequence, again, this could be a pathogen of interest, and then another non-target, so like a closely related fungal species or something. And so if you kind of move down that sequence, you'll see these arrows that indicate nucleotides that differ across the alignments. These are the genomic regions that we use to design primers for amplifying the pathogen and making it so we can distinguish it over other potential non-targets that are in our sample that we're testing. And so typically once we have the primers, we develop a probe that has fluorescent tag on it. So then we can use light detection equipment to help us out with that for diagnostics. So to kind of go through real shallow scale our diagnostics that we use, essentially we collect a sample. This could be, for example, honeybees that we use for pathogen testing. It could be soil. It could be the plant itself with lesions on it. We extract the nucleic acids. So we get RNA and DNA indicated here. What I'm showing you here is this red helix is just showing you the pathogen DNA. Typically, when you're doing this type of extraction, you're going to get a lot more of the bee DNA than you are the pathogen DNA, relatively speaking. And so I'm just kind of showing you that with the colors here. But then you want to preferentially amplify the pathogen data, and that's what we use PCR for. So to kind of give you a little bit more details, this is an extraction, a hypothetical extraction of bees, a bee sample, and we're interested in knowing whether there's 2 pathogens there. And so the extraction here shows 2 pathogens are present. You'll notice that pathogen 2 is in lower, relatively lower concentration than pathogen 1 is, given the red and blue helices there. And then we amplify and use a probe-based assay. So if there's amplification of those targets, it lights up for light detection equipment to tell us that. And so we can multiplex. So we can, in a single test tube, we usually test for 3 different pathogens. Here I'm giving you an example of 2, so we call that multiplexing. This is an output that we get. So fluorescent signals on the y-axis that can be low or high, and then the amplification cycle for the PCR. And so if you have a sample that doesn't have the target in it, you'll get a flat line. However, if one of the targets are in there, you'll get this sigmoidal curve. For example, this pathogen 1 here, and then pathogen 2. And so you can give these different markers different probe or fluorescent tag colors, so you can differentiate them in that single tube. So from this initial analysis, what you can do is find the cycling threshold, or the CT value. It's sometimes called CQ value as well. And again, this is kind of the same graph or figure that I was showing you before, but what it also includes is this threshold that gives you the CT value. And so for pathogen 1, the CT value is 14. And for pathogen 2, the CT value is 30. So you can take the cycling threshold numbers and apply it to a standard curve. And so in a different test, you set up a set of dilutions of known concentrations of the target, and then you amplified it and tracked the CT value to make these 2 standard curves. So then you can take, for example, pathogen 1, which has a CT value of 14, bring it over to your standard curve and follow it over to get your pathogen quantity. That's on the x-axis here. And so by doing the standard curve analysis, you not only have the relative concentrations of pathogen 1 versus pathogen 2, but you can give it a number, or a pathogen load, which is the terminology that I'll use in a little bit. So we all do this in a high throughput framework. So we're a high throughput testing facility. So we do this in 96 well plates. So we can extract in these 96 well plates, and then we can do the PCR in a 96 well plate. Now, if we've had plenty of coffee and we got that nice steady pipetting hand, we can do 384s. And so that's essentially 4 of those 96 well plates in a single plate. And then we have light sensitive detection equipment that gives us those sigmoidal curves if the pathogen or target is present. And so you kind of see that on this computer right here. And then the 96 well plates being ran. So that's a little bit of the background of kind of the technology that we use. I'm sure it's kind of a refresher for a lot of you, but I just wanted to kind of explain that before I jumped into the 3 research projects I wanted to talk about, because they all use that PCR and quantitative PCR technology. So I'll be talking about bees, crops, and weed projects. So when we first got started in 2016, honeybees was one of our primary efforts to help the beekeeping industry. North Dakota actually has been the #1 honey producing state for the last 13 years. And so bees are big business in North Dakota. So what's one of the problems, and perhaps a lot of you have heard about that, is that there's been this crisis of colony declines, both in the captive bees, honeybees, and also in wild and native pollinators. My focus is on the honeybees that are used in agriculture. But if you look in the literature, you see, a lot of emphasis on trying to figure out what's causing these bee declines. And it's not just in the US, it's happening in Canada, it's happening over in Europe. And so one of the components that is of interest is understanding how microbes, how pathogens are impacting these colony declines. So before I get into the pathogen data, I just want to point out that it's not as simple as just measuring pathogens. Colony loss is a complex trait, and there's a lot of different factors that come in that can impact these colonies. So it's not only pathogens, it can be nutrition. So what native pollen sources there are, or artificial food sources the beekeepers are giving their hives. Weather, climate is a big issue. So in terms of weather, more immediate weather patterns, severe storms, snap of cold, snow, things like that, those extreme weather events that happen with climate change can impact these bees. Pesticides, so there's a lot of pesticides, insecticides that are used on crops that get their way into these bee colonies. The way beekeepers manage can impact survival. The genetics of the bees as well. And then mites. So mites are kind of a vector for a lot of the pathogens I'll be talking about. And so these are important to also look at, these varroa mites. And I kind of bring them up during this project that I'll talk about. So there will be a quiz. These are all of the viruses and other microbes that we test with our pathogen panel. There are 15 total. And what you'll notice, and just quickly point out, is that most of them are viruses. On this left-hand side, we do detect a couple bacterial pathogens that cause foul brood, and then 2 fungal pathogens as well. And so not only can we detect them, but we can also quantify them. So for this project that we wanted to work on, we knew that there was a lot of movement of bee colonies across the US. And when you're moving a lot of colonies or moving any type of animal, this can introduce pathogens and other factors that can impact their fitness. And so we wanted to kind of take a look at these migration routes, or what I call migration routes, transportation routes, that are happening in North Dakota beekeepers. And so when we did this survey, it was actually surprising and quite amazing what these beekeepers do with their hives. They'll move on. There's one migration route. They start in North Dakota, go all the way to Florida for pollinating citrus trees, and then go all the way over to California. So just a large transportation route. What you'll notice though is there's a pattern here. So there's a lot of beekeepers going from North Dakota over to California. And you might be wondering why that is. In fact, 2 million hives. Starting now, actually, February is kind of the key time that a lot of these beekeepers move their bees to California. And it's for these trees, for almond pollination. 75% of the world's almonds come from the central part of California. So almonds is a big business, just like beekeeping is, because we know that bees help pollinate these trees and help boost yield for these almond growers. So that's why all these bees are moving into California. And so our research question was, how is colony strength, so an indicator of colony fitness, is associated with pathogen loads in these migrating honeybees? And so we didn't want to spread the data too much, and we focused on 3 main migration routes that a lot of beekeepers use, and that's indicated here in this panel. So there's a direct migration route. There's this route that takes their bees to Texas, then over to California, and then there's one that they'll bring their bees over to Idaho for storage in potato facilities during the winter. So we looked at these 3 migration routes. And then what we did is we slapped stickers on these research hives, and we went out there and sampled once in California and twice in North Dakota for 2 years for this study. And we collected at each of these visits, colony strength was estimated, varroa mites. So we checked how many mites there were per 200 bees. And then we took a sample of those bees for this pathogen testing. And so this was a project in partnership with the North Dakota Department of Agriculture. And so I just wanted to share this picture because I've made the mistake of saying bees are migrating, and a lot of people get a picture of these little bees migrating to California, which would be amazing. And it's not unheard of, you know, with monarch butterflies, but this is how they get there. So they just stack their colonies on truckloads, about 400 colonies strong, and then they ship them. And they net them over so the bees kind of stay in the hives and on the trucks. So that's how they get them there. And so whenever you do field work, you have to show some pretty pictures. So here's the landscape we were in California, central part of the valley in these almond groves. And then in North Dakota, much different habitat. It's, mid to late summer, and we're usually around CRP, native prairie grasslands for the most part. And of course, whenever you're trying to take a picture with bees around, you always get bee bombed or bee photo bombed. So there's always a blurry image of a bee in these pictures. All right, so this is what a frame looks like. So we pull out these frames to assess population strength or colony strength. The queen is actually on the slide. So I'll give you a moment to kind of take a look. I could start singing, one of these things is not like the other. So you should be able to find her. But essentially what we did is we wanted to quickly assess what these colonies' strengths are. And so we would pull out 10 frames, look at how many bees there were on the frame, and use a standard method of, if the frame was 2/3 of the way full, we would count that or check it off as, a frame to count in the strength estimate. And hopefully you had enough time to look and find her. She's right there, right on the top. So you can see she's got that elongated abdomen. So for our bee testing, we would not take bees from this frame. We would pull out another frame and collect worker bees for that test. Because you kill the queen, you kill the hive. You don't want to do that. And of course, the girls get really angry when you open up their hive. And so they came out and attacked us quite frequently. That's why you wear these bee suits. So I thought this was an interesting picture. They're all over my colleague here. And then just another picture. What's happening right here is my lab manager, Megan O'Neill, she's sampling the bees for varroa mites. And so we do an alcohol wash for that. Those bees that are euthanized are actually the same bees we use for our pathogen testing. And then you get it back into the lab. So we have liquid handling system for our DNA extractions. And then of course, a picture of me doing the PCR setup. And when you're running so many samples, you get a little bored. So you got to throw on that bee suit and see if you can do lab work. Usually it doesn't really work that well, especially with the hood on. You got to take the hood off. But we try to have a little bit of fun in the lab too. All right, so let's get to the data. So we have data from 8 operations, 8 commercial beekeepers. And so their operation ranges from having 2,000 colonies to 20,000 colonies. So these are large operations. We have a total of 957 sampling events across this data set. And for the variables that we included in our modeling, we have colony strength at visit, we have colony loss if it was known, migration route, and then pathogen diagnostics. And so if you do the quick math, you will find out that we pretty much did about 14,000 individual pathogen tests for this. So here's some of the data. So this is the pathogen diversity estimates across the entire data set. So on the y-axis, what we have here is a number of colonies with a particular number of pathogens in it. The number of pathogens is on the x-axis. And so what you'll see here is the mode, or the most frequent diagnostic in terms of pathogen diversity, was 6. The average that we found was 5.3 pathogens per colony. And so what was kind of a surprising thing is that a lot of these colonies have subclinical disease in them, or at least have the pathogens that we know cause disease. This next table is showing you the prevalence of the top 7 pathogens that we found across all the visits. I told you there was going to be a quiz, what these all are. But the first one, just a fungal pathogen. So what was surprising is 95% of the colonies that we tested had that pathogen. And then the remaining of the top 7 were viruses. Deformed wing is a serious virus. But I want to point out these 2 viruses that are highlighted in red that were found in about a quarter of the visits because they become important in the modeling and in the analysis. So these are what kind of came out of our analysis for the study. So we controlled for resampling hives. I'm sparing you from the modeling piece of this, but these were the important bits. So the first was we found those 2 pathogens that I highlighted in red. Associations with those 2 pathogens was that if they were at higher loads or higher densities in colonies, those colonies tended to be weak. So they had low population sizes. We also found one of the migration routes kind of fell out that had weaker colonies compared to the other 2 migration route types. And then of course, there's always this seasonal or annual fluctuation that you have to incorporate in models. And so there was a year impact. So we did have stronger colonies in 2020 than 2019. So that came out of the analysis. And then in a separate model, we were interested, okay, we're doing all this tests for 15 different pathogens. How does the diversity, which I define as the number of pathogens per colony, how does that relate to colony fitness? And so we found that there was, in weaker colonies, there was greater pathogen diversity. And so we're going to take that back to our clients because we have a lot of clients that send us B samples. Now we have these indicators that we can focus on to help our clients. And so some conclusions for this is that we found most colonies have subclinical infections. So we found a lot of different viruses in these colonies during our research project. We found weak colonies were associated with the densities of 2 viral pathogens, and there was greater pathogen diversity in weak colonies. And that transportation route, those routes that beekeepers take have an impact on colony strength. So it's something to kind of think about with these beekeepers. And the whole point of looking at pathogen levels is to see if it can be a proxy for colony health and colony persistence. And like I said, we do have clients from across the US that provide samples for our testing. We've tested over 2,000 samples in 22 states. So we have a lot of different projects going on, as well as individual beekeepers sending us samples. So I'm going to shift a little bit. Instead of talking about bee pathogens, I'm going to talk about crop pathogens. So for crop disease, pathogens and pests contribute dramatically to worldwide yield losses. There was a study that just came out in 2019 that looked across the 5 staple foods globally, 5 staple food crops globally. And if you look across yield loss, these values are kind of the mean yield loss associated with pathogens and pests. So these were evaluations from experts across the US and globally. And so when we define pathogens, we're talking typically microbes, so bacteria, viruses, and fungal pathogens. Pests can be nematodal worms, they can be insects, they can also be considered weed species. So that's encompassing all of those. But what's concerning is that there's a lot of yield loss because of pests and pathogens. And so one of the important first steps in solving these issues is diagnosing when you have a problem, when you have a crop disease. Some of the trouble is that symptom-based diagnostics in the field is difficult and can cost growers money. For example, take these 2 corn pictures here, A and B. You'll notice that there's these streaky lesions found in both of these pictures. One is caused by a fungal pathogen. The other is a bacterial pathogen. Now, if a grower was to say, oh, I have a fungal pathogen, a fungal disease, I'm going to spray fungicide in my field, and it's actually a bacterial species, that fungicide was a waste of time, waste of money, and potentially just introduces that fungicide into the environment that could lead to fungicide resistance. And so it's important to get diagnostics right. In saying that, there's a lot of great visual guides out there that expert pathologists have put out to help growers identify fungal disease versus bacterial disease. But they always have these caveats that I'm showing you here. You know, symptoms may look similar in other common diseases, sometimes causing confusion and misdiagnosis. Symptoms may vary in hybrid susceptibility. By hybrids, you know, if you have variety of corn 1, variety of corn 2, the same pathogen might show a little bit different symptoms in each of those. And so it's a complex disease sometimes to diagnose. Now, there's of course laboratories that help growers out with this diagnostics. A lot of it is relied on traditional lab techniques. So using a microscope to look at the spores or the fungal hyphae or mycelia underneath the microscope to help identify not only it's a fungus, but also what kind of fungus, what species. You can also, of course, culture these, but as this picture shows, if you start getting a lot of samples in, you start taking up a lot of space in terms of culturing. Also, you're assuming that the pathogen that you're detecting or the isolate you're detecting is culturable. Some of these pathogens only grow on living host tissue. And so there's some trouble with diagnostics in that respect. Like I say, there's a fresh sample is required too. And what's important is that you can't test environmental samples. So you can't test soil necessarily using a culturing method, right? You can't test the air. You can actually test the air with a culturing method. But again, that's assuming you can culture that pathogen that you're looking for. And so there needs to be additional diagnostics to help you find that pathogen in the environment. And so this, of course, is important because in terms of surveillance, a lot of these models that are used currently are based off of weather data and disease that is found in the region or in the area. The problem is when it's linked to disease, you already have a problem. So any type of management you're doing is going to be reactive and not necessarily proactive. Now, weather data is typically used for these models because it's the easiest to collect. However, I want to point out it's only one side of the disease triangle. So whenever you talk about disease and pathology, all 3 sides of this triangle have to be present for disease to show up. And so when you're looking at weather data, you're really only looking at environmental data. So you're looking at, you know, the temperature, precipitation, drought, things like that. Incorporating the host plant susceptibility, you know, the variety will help improve these models. But what we're interested in is helping to improve the other side of the disease triangle, and that's amplifying and detecting as well as quantifying the pathogens to increase the resolution of disease risk. And so new to 2020, there's this National Predictive Modeling Tool Initiative that our research group helped lead that currently has 27 collaborating universities and research institutions involved. And essentially what we want to do is kind of complete that disease triangle for more proactive management tools, these forecasting tools. So we want to provide inputs for big data processing and modeling to occur so we can have these risk models, these forecasting models for growers to use in more real time. And so our job at NAGC is to develop these molecular tests, these diagnostic tests to sample the environment. And so we get in soil and residue samples. So residue, if you're not familiar, is just degrading crop material that's left over from previous seasons. Typically, there's a lot of pathogens that overwinter on crop residue, not necessarily just the soil. So we test both soil residue and then spore sampling as well. So we're providing these pathogen inputs to include with weather as well as field history data, crop data, to help increase the accuracy of these heat maps. So this is an example of a blight risk map that's put out for wheat that is based off of weather data. So it's showing you areas that there's increased risk of this fusarium blight, a fungal blight in wheat. And so we want to help improve these heat maps with this National Predictive Modeling Tool Initiative. And then, so lastly, for my last 5 to 7 minutes, I wanted to jump into weed diagnostics and what our lab is doing with that type of pest management. So herbicide resistance is an increasing issue. What I mean by that is that there are weeds that are escaping herbicide applications in row crops. And you can kind of see this field right here has a high infestation of weeds. Weeds, a simple definition, is a plant that is growing where you don't want it to. And so this picture is illustrating how it can be a big problem in row agriculture. And so unrestrained weed growth has been estimated to reduce crop yield by 50%. So weed interference is a big issue, and it's a big problem that needs more work performed on. And so we focus on a certain species or certain group of weeds called amaranths or pigweed species. They're prolific seed breeders. They'll cast out hundreds of thousands of seeds. So a single individual can cast out a lot of progeny. They're dispersed by wildlife, flooding events, and also human movement. So when a field gets harvested and they pick up these seeds, they can transfer it and contaminate other fields. So weeds can move via that method. They have a very fast growth rate. This picture here is an NDSU researcher standing by Palmer amaranth, which is a pigweed species. I think this one's like 7, 8 feet tall. They can get upwards to 10 and grow 4 inches per day. So again, they can outcompete these row crops, and they can lead to 78% yield loss in soybeans under heavy infestation. And we focus on amaranths because there's a lot of herbicide resistance in this group of weeds. There's single individuals that have been found to be resistant to 5 to 6 different modes of action of herbicides. And so it's a very important active area of research. So how do you know if these weeds are herbicide resistant? Well, you bring them into the lab, you grow up the seeds, you spray them, and then you see the outcome. And so this is showing you a greenhouse bioassay doing exactly what I just explained. And you'll notice that these ones that are growing pretty well, these are the happy, healthy, herbicide resistant ones. This other image here is showing you some herbicide damage, having this decaying leaf here. So these bioassays are great, critical, still important to run to find herbicide resistant weeds. They take time though. You have to grow up these seeds. As they grow up, you got to spray them. And so the nice thing is that there has been some genetic markers that have been found associated with herbicide resistance. And so I just want to bring you through one of those that we're currently working on at NAGC. And first, I want to kind of talk about the genetic basis of it. So what I'm showing you here is an alignment of two nucleotide sequences, 2 DNA sequences of a portion of this PPX2L gene found in Palmer amaranth. There's a wild type sequence on top and a mutant or a resistant genotype on bottom. And so what I'm doing here in this first red circle here is I'm zooming in on an exon. So this is the exon zoomed in. You'll notice that there's a gap here, and then I zoom it in here towards the bottom. This gap in the mutant sequence causes those individuals that have that mutation, that deletion, to have a change in the protein product that this gene makes. And so we call this mutation the delta G210, because what happens in this mutant sequence is that there's a loss of this glycine amino acid. And so we know from general biology, structure equals function. And so when you mess with the amino acid sequence, you're going to mess with that function and as well as that structure of that protein product. So these are hypothetical protein illustrations. And so we know how this particular herbicide, PPO inhibiting herbicide, works, the active site. What it does is it competes for the actual substrate that this enzyme in the weed breaks down. And so what happens is that in the wild type, there's only space for that herbicide molecule to compete. And so it shuts down that pathway. However, in the mutant, it's still a functional protein, but that gap that molecule, the herbicide molecule once sat, is a little bit bigger. And so now it's not as effective. And so we can see this at the genotyping level, or we can at least find that mutation. And so that's what we're doing in the lab for 2 species of pigweeds, Palmer amaranth and water hemp, for this Delta G210 site. What we have is archived samples. All we need is the DNA sample to do this genotyping. And so this is just a quick output showing you the output of the piece of equipment that allows us to genotype. And in our archive samples that were collected in North Dakota, collected in Minnesota, we're finding these resistant or mutant genotypes in the populations. Generally, there's a lot more that are the wild type, but we are finding indication of these herbicide resistant plant populations in some of the species that we've already tested for. So right now we have an upcoming survey. We're going to do more finite work with weed scientists at NDSU, as well as those across the state, to develop a distribution map. So not only do we know where water hemp, for example, is found, but we want to include a layer in this map where there are herbicide resistant populations based off of the genotyping that we're doing. And we're doing other herbicide resistant traits, genetic markers that are linked to herbicide resistant traits, to develop kind of more of these layered maps. And again, this is just to help producers avoid that application of infective herbicides. So if I can wrap everything up, the point I want to get across is learning these molecular protocols create a career opportunities that span disciplines. I started in wildlife, but a lot of the technology I learned there is being currently used in the agricultural sector as well. PCR-based tests, even though they've been around for decades, they're still pretty functional and answer a lot of pressing questions in disease and pest management. And there's a strong need for a workforce in agricultural diagnostics. You don't need a high-level degree, a Ph.D. to do the bench work. You know, we need 2-year professionals, 4-year professional degree students and technicians to help with a lot of the lab work, because pests and pathogens are not going away anytime soon. So with that, thanks for the invitation, Innovate Bio. Thanks to all you for listening. I will put in the chat these 2 websites, and as well as my email. I'm happy to answer any questions offline, but I think I have some time for some questions right now. So thank you once again.
Hi, Zach. Just to make things easy for you, here's a couple of questions that have turned up. One is about your bee project, and it says, it asks, do you see any difference for pathogen diversity between, sorry, it just moved, between small and large beekeeping facilities?
I would love to have a data set of hobbyists. I would guess that there would be differences between hobbyists, you know, that have 1 or 2 colonies, and these commercial beekeepers that move their hives all around the place. I know there's been research out there looking at that, and how beehives that have been elsewhere brought into an area have infected, or at least there's a correlation with infections going from these migrating hives to these what you could call clean hives or non-migrating hives. Because there's a lot of intermixing going on with bees. They can travel 2 miles to find pollinating sources. So the short answer is, would there be differences? I would expect it, yes, but we personally don't have any data to support that.
Here's another thoughtful question. It's a number of questions. Did you compare transported bees to non-transported? What is known about the effect of transportation on bees? I'm thinking that there's a lot of vibration in transported bees, and wondering if that contributes to weakness, susceptibility to pathogens, colony collapse, or just the effect of the vibration.
Yeah, so honeybees did not evolve going 90 miles an hour down the interstate. So definitely transporting these bees can be stressful, especially, you know, when you're transporting them to California where there might not be food available immediately. So yeah, that's definitely a component of that. I'm sorry, what was the second part of that question?
The main thing is is there any evidence that the vibration would contribute
Oh,
pathology or weakness or susceptibility.
yeah. Yeah, it would totally, it could be an impact for sure. Yeah.
I'm not sure which part of your talk this refers to later on in your talk, but the question is, how would you find a genetic marker?
The great thing about being in a diagnostic lab, we have labs and research groups like Dr. Ware was talking about that does all the genomic sequencing analysis for us. And then a lot of times what happens is that if there's an unknown nucleotide sequence that's available, you can compare to another well annotated genome to find out what that particular region or nucleotide sequence matches to. And so that's kind of how pathogen testing goes too, is that you can find closely related species to compare sequence alignments to. So a lot of what we do is we let the foundational and academic research labs find that information, and then we grab it and we use it in some sort of practical way. So a lot of that stuff has already been done before it gets into our hands.
You partially answered that, but here's a more specific question on markers. You talked about genetic markers for herbicide resistance in weeds. Do you know how that specific genetic marker was found?
How that was found? Well, the mode of action or the site of action for these herbicides are known for some of them. And so when you know the mode of action, then you know kind of the protein that it's targeting, and then you can trace it back to the genome where that gene is in. And then when you do the sequencing and you do those alignments like I did, there's a big aha moment that, whoa, there's this functional protein, or most likely functional protein, that it has this deletion that didn't start a stop codon prematurely, but there's this deletion. How is that impacting it? And then you can do these crosses in the lab, greenhouse experiments to confirm that genetic marker is associated with that phenotype.
Okay, here's an issue-related question. Roundup resistant pigweed has become a real problem due to the massive amount of Roundup sprayed on the fields. This is a man-made problem which is now hard to contain. Is there any serious research to use natural methods of pest or weed control in agriculture?
Anything that I would say would be kind of my opinion on that, because we're not actively working on that. So I don't know how much to comment. There are, biostimulants, there's adding certain inert material to help with those issues. That's something we're not really working on, but I know it's out there. Because the problem is that a lot of farmers have a limited amount of chemical control options available, and weeds are getting pretty smart and by smart, I mean, they're evolving resistance to a lot of these chemicals. And so what's best is to hit them with a lot of different things and not the same thing over and over and over again. Because if you start doing that, then you start getting these resistant problems.
Yeah, probably in agriculture, they should practice what's done in cancer treatment, which is to hit with two agents at the same time. And then the chance of evolving resistance to both at the same time is a product of the two individual events, much harder.
Yeah.
Before I forget, in your chat box, there is a link to a survey about Dr. Bateson's talk. And we would really appreciate it if you would follow that link and take that very brief survey. We do have time for a couple more questions. We've got a number of comments about what a great talk that is. From my point of view, I really like the fact of how your career, built upon your interest in nature and then wound its course in a idiosyncratic way. And I think especially, students, that's a great lesson because we don't always go in a straight line to where we end up in life.
Yeah, and it's been a fun trip.
Well, Zach, I guess we'll take a couple minute break for our next talk. And thanks so much for joining us today.
Yeah, thank you once again, and I'll put my email and stuff in the chat.
One more question came in.
Oh.
I am a small commercial beekeeper. Do different climates play a role in the pathogens of honeybees?
I'm sure, absolutely. I mean, weather is going to be a big component to that. Bees that stay down in Florida are going to have different pathogen distribution and types, than those found in North Dakota. So that's definitely something you need to include in these models when you're trying to pull out what's important with colony loss.
Here's another one popped up. Would it be possible to alter a gene in the cells of a pigweed to help them resist things like Roundup and pathogens?
Yeah, and there's actually there's research going on right now at North Dakota State University where a research group is trying to put susceptibility back in through gene drives. They're really at the early stages, but we're really excited about watching that progress because if we could throw it back in, change the sex ratios of these plants too, where they only produce male seeds, because some of these are dioecious weeds, that would be a phenomenal approach to weed control.
Zach, do take a moment to look at the chat box because there's a lot of nice compliments to you. Here's one more question I think we have time to answer. Have you commercialized any of your tests?
Yes, so that's the one nice thing about the lab I work for. We're a not-for-profit. All of our tests are available to anyone that wants to use them. It's a fee for service. We try to find ways to make it affordable for beekeepers, for farmers and growers. We also are interested in education resources too. So just like Innovate Bio, we're interested in reaching out and interacting with teachers and students. But yeah, our testing services are all available too. So.
Great. Thanks, Zach, for joining us today.